Hey everyone, welcome to this demo on AI in drug discovery. Do you know that it could take as many as 12 to 15 years to bring a single new drug into the market? What's more, only 14% of all the drugs end up with approval from the FDA. The reason not necessarily being the efficacy of the compound alone, but also around operational delays, uncertainties leading to dropouts, and so on. Introducing Relio's AI in drug discovery, a suite to expedite the drug development process through ML and AI. Clinical researches are conducted across globe, and the recent past has witnessed a tremendous expansion of the clinical trials market into diverse cultures and languages. Organizations that conduct clinical trials outside of the United States, governed by guidelines and practices, face the need to translate a range of documents like clinical protocol documents, case report forms, patient informed consent forms, so on. Now, translation of these documents becomes critical and mandatory for any clinical trial being conducted on a global scale. This use case uses medical translation service from Azure to automate the translation process. As you can see, once we upload a document, say in Chinese language, it hits the Azure service and lets us download the translated file like so. You are now able to see the translated document, which can then be used to extract other important information. The next step in order to utilize the information in these documents is to extract them automatically. We make use of the Azure Health Text Analysis service in the second use case to extract named entities, relations, assertions, and standardized notations. These in turn find use in other places like identifying patient cohorts, building knowledge graphs from millions of documents, which is otherwise next to impossible to be done efficiently. As you can see, the solution recognizes the various entities, like say, for example, the medication name or the medication route, and so on, along with relationships. Say, for example, fluorosil here is actually related to the entity 24 hours through the time of medication relation, and so on. The third use case here is on building a named entity recognition model for custom biomedical entities. This was trained using Spark NLP state-of-art deep learning models and medical embeddings, which can then be used for other purposes like building knowledge graphs and so on. Now, the highlight of this solution is in the way we had automated the generation of the labeled data or the annotation data for the NER problem. We made use of pre-annotations and active learning strategies with minimal human involvement to build the final model. Here, it is able to recognize the drug as well as the route of administration from the input text found on the left side. The drug efficacy determination solution is an ML-based regression model to determine the bioactivity of a drug candidate in terms of IC50. The half maximal inhibitory concentration, or the IC50, is a measure of the potency of a substance in inhibiting a specific biological or biochemical function. The chemical compound is represented using a SMILES notation as shown here, which is then converted into molecular descriptors through packages like RDKit. Now, the numerical values of molecular descriptors are used to quantitatively describe the physical and chemical information of the molecules. For example, 1D descriptors could be count or the type of atoms in a molecule and the molecular weight and so on. On the right, when we upload the molecular descriptors, what we find is the value of the IC50 corresponding to the molecule for a particular target. Here, in this case, it was determined for a target associated with Alzheimer's disease. Now, this solution drastically reduces the time spent in screening the correct compounds, which can be taken up for further trials. The last use case in this demo is based on image data, which specifically finds use in clinical trials that have image endpoints. Here we have automated the detection as well as counting of blood cells like red blood cells and white blood cells and platelets using an object detection model. Now, with a solution like this, uh, reduces the effort and the time spent by doctors and lab personnel by automating this complete process and helping in the diagnosis process. I hope this demo has been useful. Thank you so much.